Drones appreciation. I think we've saved the best for last. Probably many people know Claire. And she produced her first bird book while she was still at school at Westerford. And she came to UCT and did her undergraduate at UCT and then did honours at UCT.
Thanks very much. Thank you, and good morning. Are you all able to hear me? Is the set about right? Excellent. Thanks very much. Just before I start, could I please mention that I have a small stammer that sometimes unpredictably becomes a larger one. If it does, please don't worry. I promise I'll get there eventually. Well, thank you very much for making it through until the end of the week and being here this morning. It's a real pleasure to have this opportunity to share with you something of the lives of some wonderful African birds that have intrigued me for the last decade or so. So, as Peter said, my interest is principally in interactions among species, such as parasitism and mutually beneficial interactions. And species interactions such as these are the background to all of life, and I would argue that they help to explain why life is as thrillingly diverse as it is. And by way of this story, I'd also like to underline how interactions among humans in all of their diversity have helped me, for one, in trying to answer these and other research questions. And for me, that began, as Peter said, in childhood, when I had the great good fortune to grow up just down the hill from the Fitzpatrick Institute, where I used to cycle up after school and bother the likes of our current director, and here we are together with Callan Cohen processing a netted cholo leafy on Mount Namuli in Mozambique. I was also fortunate enough to be partially brought up by the Cape Bird Club, including several kind people in this audience. And although I didn't, of course, appreciate it at the time, these early birding experiences laid the foundation for all of my subsequent research. I went on to study here, as you heard, followed by a PhD overseas. Yes. Ah, I'm sorry. Sure. Perhaps if I... Is that better? I'm just turning down the knobs here. Okay, take it up a notch. All right.
And the volume, please.
Over here?
Can you button these buttons, please?
Sure. Is that any better for you?
We found it's better without that.
Really? I can try. I'm rather soft-spoken, but I'll do my best to bellow. Is that better? Okay, good. Shall we try again? Okay. Thanks very much. Thanks for your help. So yes, fortunately, all you've missed is the preamble, which is perhaps the least interesting. So just to reiterate my good fortune in growing up just down the hill from the Fitz and close to the Cape Bird Club, who provided me with many formative experiences that underpinned all of my subsequent work. As Peter said, I then went overseas as a postgrad and for my sins worked there for some years after, and I'm now very happily back home since 2016, although I still work part-time in both institutions and regularly move between the two of them and between my field sites elsewhere in Africa. So let's start in Zambia in the dry season, peering down at this clutch of eggs that's just been lowered down to us from the nest of a fork-tailed drongo. And in it lies a battle over identity, much as in immune systems and in computer security, and these days in our world of fake news. So is there a cuckoo egg in this nest? And if so, which is it? Well, host parents too find it hard to tell. They often overlook a cuckoo and they pay a high price if they're tricked. The young parasite throws all of the host's eggs out and the parents waste all their investment raising the wrong chick. So cuckoos, of course, are brood parasites, the cheats of the bird world that exploit the care of other species to raise their young, and in so doing sidestep the costs of parenthood. And to get away with this, cuckoos are master tricksters. This near-perfect egg mimicry is the outcome of coevolution, which is the process through which two or more species reciprocally influence each other's evolution. And work, especially on the common cuckoo in Europe, has been instrumental in showing the power of coevolution to shape beautiful adaptations such as these in nature. So the process kicks off when host species start to defend themselves against parasitism by removing foreign eggs from their nests. And cuckoo eggs, which are by chance a better match to host eggs, are more likely to evade this, which over generations leads to the evolution of the amazing mimicry of host eggs by cuckoo eggs that we've just seen. This in turn selects for yet better defenses from hosts, which in turn selects for better trickery, and in theory the two species can thus become locked in a coevolutionary arms race, each to stay one step ahead of the other. Now such reciprocal interactions don't only involve enemies like parasites and hosts or predators and prey, they can also involve mutualists, species that benefit each other, such as plants and their pollinators, or perhaps the symbiotic microbes that are influencing our health from within our guts right now as we sit in this room. So what all these interactions have in common is that they involve a nimble and ever-changing partner. So as you adapt in order better to exploit or to defend or to cooperate, so the other party adapts in return. And as John Thompson puts it, much of evolution is about staying in sync with prey, hosts, or mutualists, and staying out of sync with predators, parasites, or competitors. So what this means is that even if the physical world were to remain constant from now on, this process would still continually drive evolutionary change. Now, it's ever clearer that coevolution has long reach, not just through biology, but also through society, since it also underpins our own human constant battles, with, for example, a rise of resistance against antibiotics and pesticides, and the decline of crop pollinators, which are all reminders that we too are subject to the forces of evolution playing out on an ecological stage that we ourselves are ever modifying. So in a world full of such pressing problems, why should we study cuckoos? Well, I'd argue that a fascinating history of cuckoo research has shown us that they give a wonderful opportunity to unpick the mechanisms underlying coevolution in the wild, where we can observe real selection pressures and test them with field experiments. But cuckoos are not the only cheats of the bird world. About 1% of all the world's birds are brood parasites, and brood parasitism has in fact evolved 7 times independently in birds, as well as in various insects and fish. So it's evolved 3 times independently in different independent groups of cuckoos, shown here, once each in the parasitic finches of Africa and the honeyguides of Africa and Asia, the cowbirds of the Americas, and in this rather peculiar South American duck. But like most of life, most brood parasites turn out to live in the tropics, where climatic conditions have been relatively stable over evolutionary time. So perhaps then tropical species might give us a different window into what coevolution can do when it's been given more time to run riot. So in this talk, I'd like to stay right here on our home continent of Africa. We're going to start in Zambia, a country blessed with almost a third of the world's brood parasitic species, and focus especially on 3 of these 7 families of cheats: the old world cuckoos, the honeyguides, and the parasitic finches, each here being fed by some of their respective hosts. We'll then travel on to East Africa, to Mozambique and Tanzania, where thanks to the Jekyll and Hyde qualities of one of these species, the great honeyguide here at bottom left, we'll consider how mutualistic rather than parasitic interactions might drive biological diversity of another kind. And in so doing, I hope that this cast of heroes and villains will give us an African snapshot on how all of life is confronted with the constant need to outwit enemies and to cooperate with partners. And we'll see, I hope, how this ever-changing race can drive diversity among individuals, among populations, and even among species. Now, the reason I study cuckoos in Zambia in particular is the legacy of this extraordinary man, Major John Colebrook Robgent, who farmed there until his death and was a voracious but very meticulous egg collector. And I first heard about him when I was a PhD student 15 years ago and drove my ancient bakkie up to Zambia to meet him. And I won't ever forget him standing on the doorway of his farmhouse deep in the bush and greeting me with the words, "Ah, Spottiswoode of Berwickshire," And he first showed me, apparently that wasn't a fatal mistake, so he first showed me that it might be possible to study these birds, which I've done ever since, and I'm very grateful to him for that, and for welcoming me on his farm and in his beautifully documented egg collection. Now, slightly more cheerfully, perhaps, everything we do in Zambia also relies on this fantastic team of field assistants from the local community, who every year find over 1,000 nests for us to use in our research. And they're genius field workers in an age in which it can be easier to sequence a genome than to get the sample that contains it. So, for example, African cuckoo eggs that we saw at the start came from this drongo nest just here. And this is one of many photos that I don't show to our departmental health and safety officer back in the UK. So we've seen already that African cuckoos impose major costs on their hosts, but the other species that we study are also highly virulent brood parasites with other specialized adaptations to kill off the host's own young. So, for example, honeyguide chicks hatch already equipped with these needle-sharp hooks at the tips of their beak, with which they use to bite pretty much anything in close proximity. But perhaps because their hosts all breed deep inside dark holes, no one had yet seen what they do with them in host nests. When we planted infrared cameras underground, this is what we saw. So the big chick here is a 3-day-old honeyguide, and the small chick alongside it is a swallowtail beta that's hatched about 20 minutes earlier. And those eggs in the foreground will soon hatch too and be picked off one by one. Now, we filmed this in a number of nests and found that host young can take anything from 9 minutes to over 7 hours to die. So unless you enjoy this kind of thing, I'll move swiftly on to the conclusion that hosts pay a high price for being tricked by a brood parasite. They lose all their own offspring, and they waste an entire breeding season tending a giant imposter. So, for example, by the time a honeyguide chick fledges, looking like innocence itself, it weighs the same as an entire brood of bee-eaters, having eaten all the food that the host parents have brought to the nest. And it's these high costs of parasitism that drive the evolution of the amazingly good mimicry of eggs of many brood parasitic birds that Coevolution has shaped to look just like those of their hosts. So once such impressive mimicry has evolved, what can a host do to fight back? Well, a very prescient idea came out of southeastern Africa a century ago. This is Charles Swinnerton here in a pose very familiar to field biologists to this day, standing alongside a broken car. And inspired by the birds of Zimbabwe, he suggested that hosts could use their own eggs as defenses against parasites. So he wrote, "I doubt whether mimicry would always end the matter, for when a cuckoo's egg became indistinguishable from its hosts, variation in the latter would still afford the means of distinguishing it from the cuckoo's." So in other words, he said, hosts can make their eggs more difficult to mimic by evolving variation in egg appearance among females, or if you wish, signatures of identity on their eggs. Now, one of the groups of African birds that inspired Swinnerton was the Prinias and Sticulars, birds that I'm sure you'll all know well, such as this tawny-flanked Prinia here being held by Tom Hamusakili, the son of the tree climber whom you met earlier. And its modest plumage gives no hint of the artwork that goes on in its reproductive tract. So these eggs were all laid by different tawny-flanked Prinia females, all within a few hundred hectares of bush in the south of Zambia. Now, one female always lays the same type of egg within her lifetime, but every female is slightly different, just as we might expect if these egg markings serve as signatures of identity. And these spots and scribbles even look like human signatures, but what Swinnerton crucially realized is that they might also act like them if Prinias exploit this diversity. Oh, I'm terribly sorry. What shall we do? Shall we try without the microphone? Ah, well, interesting. Okay. Sorry, we have some issues with the sound being slightly asymmetrical.
The sound up here is okay.
But
I don't know what's happening.
sure. Shall we try without the microphone? Are you able to hear me? Okay, let's give that a go. And I'm likely to tail off, so please just wave your arms when that happens.
Please, maybe don't look at the screen while you're talking when you turn sideways.
Right, I'll do that.
Thank you.
Okay, shall we try again? So just to recap then, these eggs here were all laid by different tawny-flanked Prinia females, all within a few hundred hectares in the south of Zambia. So one female always lays the same type of egg within her lifetime, but every female is slightly different, which is just what we might expect if these egg colours and patterns function as signatures of identity. So these spots and scribbles, as you see from this example here, even look like human signatures. But what Charles Swinton crucially realised is that they might also act like them if prionies exploit this diversity to help them to recognise their own eggs. Are you all still able to hear? Great, thank you. So we can test Swinton's idea by pretending to be cuckoos ourselves. I swapped eggs among nests to simulate parasitism and then checked whether the host rejected the foreign egg or accepted it as one of its own. And what this showed is that hosts pay attention to several different egg traits. So a big mismatch in one cue, such as pattern in this instance, in this example at bottom left, is enough to trigger the hosts throwing out the foreign egg. But hosts can also integrate several smaller, more subtle cues to spot an imposter. And this is what happens when they do. So here's natural selection in action. The prinia stabs the foreign egg on the end of its beak and flies away with it and drops it off somewhere else in the bush. So Swinton was exactly right that this variation in the host eggs allows it the means to distinguish it from a cuckoo's. Hosts do recognise their own signatures. But what Swinton didn't know, but I'd like to think he would have been thrilled to find out, is that there's a parasite that beautifully forges these signatures. And this is the cuckoo finch. And here is why you might not want one of its eggs in your nest. The young cuckoo finch begs very vigorously as soon as it emerges from the egg, and the host chicks soon shrivel up and die as these two particular chicks are on the point of doing. So how does it get away with it? Well, here are the eggs of cuckoo finch in the central circle with host eggs around the outside. And you can see that cuckoo finch eggs are pretty good mimics of most kinds of prinia eggs, although their markings aren't quite as intricate. But what we really need to know is not whether they're good enough to fool us, but whether they're good enough to fool a prinia. Now, thankfully, we can now easily model how a host might see these signatures and forgeries, owing to all the great strides that have been made in understanding bird vision in the last couple of decades, given, for example, that birds have the ability to see ultraviolet light, which we, of course, lack. And for me, this was all made possible by working together with a sensory ecologist named Martin Stevens. And together we found that there are 4 traits that differ slightly but consistently between prinia egg signatures here on the left and cuckoo finch egg forgeries here on the right. So first, the background colour of the egg. Second, the size of the markings, the dispersion of the markings across the egg, and how variable they were in size. So, how variable the markings were in size, that is. So what this means is that on average, there are discrepancies between hosts and parasites. So cuckoo finch eggs are imperfect forgeries of host eggs. So our question then is whether prinias can exploit these imperfections to defend themselves against parasitism. And what our field experiments showed is that this is exactly what prinias do. So these specific cues that prinias use to reject foreign eggs were precisely those 4 traits that differed most between real cuckoo finch eggs and their own. So what this implies is first that prinias are cueing in on the most reliable information available on spotting a parasite, and second that they're of course selecting for ever better forgeries of their own signatures. So in the face of this, we should expect cuckoo finches to target host clutches that match their own egg type, since no bird can change her own egg colours. So a cuckoo finch that lays eggs with a blue background, like this one on the right, should target prinia clutches with a blue backgrounds and so on. But remarkably, what we see in the field is that they don't. They normally get the host species right, but among nests of that species, they're gamblers with their eggs, and they lay them haphazardly, irrespective of the host's particular egg signature. So they rely on chance matches like this one above to succeed and have a host accepted to fledging. Sorry, have a chick accepted to fledging by the hosts. But very often they get it spectacularly wrong, like this example down here, and their egg is soon trapped out by the hosts. So this really underlines that Swinton is right. Egg signatures are a very good defense against parasitism by increasing the likely mismatch between a host's own egg and those of a parasite. But Swinton also crucially realized that coevolution doesn't make species inherently better, only better relative to the species that they're interacting with at the time. And he went on to write that it's even imaginable that a race may in some cases have taken place between the host's eggs and those of the overtaking cuckoo. So in other words, as the arms race escalates and parasites forge host signatures, hosts should evolve new signatures that their parasites don't yet match. And to our amazement, we found evidence of this even within our lifetime. So by comparing Major Colbert Robson's egg collection with present-day eggs from his farm, we found that these olive green eggs here at top left are substantially more common right now than they were during his egg collecting heyday in the 1980s. And this makes perfect sense, since as some of you will already have spotted, this is the only colour signature that Prineas haven't yet forged, since Prineas don't lay green eggs. Sorry, cuckoo finches don't lay green eggs, only Prineas do. So just as we've required a new design of 200-gram note to keep ahead of counterfeiters, so the Prinea perhaps has evolved olive green eggs to keep ahead of the cuckoo finch. Now, in fact, the same diversification of signatures that we see in eggs is also seen in other settings where distinguishing self from non-self is a matter of life and death, such as in the hosts of insect brood parasites, such as ants, where odors diversify to make a parasitic enemy stand out, and even in immune systems, where molecules evolved in immune defense do exactly the same thing. So Swinnerton's signatures then can be chemical or molecular, as well as visual. But what all these three cases have in common is that the self has diversified to make it easier to recognize non-self, and enemies in turn evolve to copy that diversity. So the main message so far then is that parasites can drive hosts to diversify, and reciprocally parasites are forced to specialize too, as they chase host egg appearance. And in this lies a century-old puzzle, which I'd like to address now. So we're going to briefly leave the field and head to the lab to consider how cuckoos evolve egg forgeries. Now, the problem we need to solve is this. Some species of brood parasite target not one, but several host species. And intriguingly, different females of the same parasite species lay eggs that beautifully mimic each host. So these, for example, are all eggs from different strains of cuckoo finch females that live side by side in Zambia. So what we still don't know is how such adaptations remain genetically distinct in the face of interbreeding among individuals of the same parasite species, but reared by different hosts. Now, the best idea we have was proposed by Reginald Punnett, who was one of the fathers of modern genetics. And he was inspired by the discovery of the W sex chromosome in birds, which is carried only by females, just as the Y chromosome in humans is carried only by males. So birds, in other words, have slightly different sex chromosomes to our own. In birds, the females are ZY and males are ZZ, so it's the females who have the unique chromosome. Now, what Punnett importantly realized is that if the genes for mimicry are located on that W chromosome, then every daughter of a hen cuckoo would resemble her mother in the character of the eggs laid by her, no matter who her father was. So in other words, if the genes for mimicry are on a chromosome of which there's only one copy, which is carried only by females, it doesn't ever mix with a copy inherited from the father. So this should allow a mother to pass on an intact copy of her adaptations to her daughters, which in theory would allow a mimicry to stay specialized even within a single interbreeding species. But cuckoo finches compared to the cuckoo of Europe, which is by Punnett, face a double challenge. Like the common cuckoo that he was pondering, they must adapt to several different host species, besides the Prinea that we've already met. But in addition, each of these host species has this bewildering array of egg signatures among different females of that species. And this raises another puzzle. The very fact that the W sex chromosome doesn't recombine, that's to say, it doesn't ever mix with the matching chromosome inherited from the father, could become a real setback for these egg mimicry genes if a host is constantly evolving away. And that's because this is quite a slow process in the absence of such mixing. So if Punnett was right, and females just have a single copy of these genes, then parasites have to rely on genetic mutation alone to generate new egg bearers that might fool hosts. And this seems unlikely because, as I said, this should be a much slower process than the genetic recombination which occurs otherwise at every generation. So Max Sorensen and Wenfei Tong and I have been trying to answer this question using genetics. And now, while we've still no idea what genes are responsible for those host egg traits, we can use other markers of maternal inheritance to track them. So we hope that Africa's relative climatic stability over time might afford us a good chance of finding a clearer genetic trail of cuckoo finches than had been found, for example, in cookies in Europe. So we sequenced DNA from two parts of the genome inherited only from a mother. The W chromosome, of course, which is present only in females, but also the mitochondrial DNA, which is present in both males and females, but is inherited only via the egg and not via the sperm. So we all have a copy of our mother's mitochondrial genome. And this gives us information on the host origins of both males and females. So if Planet was right, then we expect that cuckoo finch offspring reared by different hosts should differ in the two parts of the genome that they inherit only from their mums. But they shouldn't differ in the genes inherited from both parents, called autosomal genes, because males are fathering offspring raised by a variety of host species. So what we found first was that the vast majority of the genome, the autosomal genes, don't differ between the host specialists. So there's no divergence at all across nearly 7,000 little bits of genome. And this is crucial because it confirms that cuckoo finches from different hosts do freely interbreed, and they aren't in the process of busy splitting into multiple species of cuckoo finch. But what was striking was that the maternal genes show an amazingly different picture. So both the mitochondrial and the W chromosome genes show that parasite specialization is tightly maternally linked. So cuckoo finches first started to specialize around 2 to 3 million years ago, when females split into two ancient strains targeting two different host genera, Prinia and Cisticola. So this means that cuckoo finches haven't ever successfully laid an egg in the nest of their own host since around the time that they shared the African savanna with Australopithecus. And since then, they've further diverged into unique and distinct female strains with specialized mimicry of each different Prinia and Cisticola species at our study site in Zambia. So this unequivocally supports Planet's hypothesis and so finally cracks the century-old puzzle. But what about parasitic adaptations to forge all of those host signatures there on the right? Are they also controlled by maternal genes, despite all the reasons why we might expect them to experience the process? Well, to our astonishment, it seems that they are. So red-faced testicular host eggs here on the right vary from bright to dull turquoise, and correspondingly, the two sub-lineages of cuckoo finches here on the left, who exploit them, lay two distinct egg types that copy those two extremes. Now, Prinia eggs, as we saw earlier, are more of a challenge since they're so diverse, but we've really seen that, at least in terms of egg appearance, cuckoo finches can copy that diversity to some extent. So, how did they do it? Well, the Prinia strain of cuckoo finches is divided into two substrains, but one lays either blue or white eggs, shown here, and the other lays either red or white eggs. And this, in fact, makes perfect sense because birds have only two egg pigments that colour their eggs in their shell glands, bilirubin for blue and protein porphyrin for red. So each of these substrains is associated with one kind of pigment only. And here, white is simply the absence of any pigment, since it's effectively the cull site of the shell underneath, which is white. And this at once made sense of everything that we'd seen in years of fieldwork. And that's because to paint an egg green, you need a mixture of pigments, as Punnett himself showed in chickens, and any 5-year-old child could also tell you. And here, what's important is that since cuckoo finch females probably only have one version of their egg pigment genes, which they're inherited from their mums, they can only lay either red or blue eggs, and not combine the two in order to make a green egg. So they'll need to wait for quite a substantial genetic change, perhaps a gene duplication, before they can produce green eggs to full green hosts. And this now makes perfect sense of our past finding that hosts with olive green eggs have become more common over time in Zambia. They're thriving because they have an unforgeable signature. So the key point is that the location of the parasitic egg genes on the female W chromosome puts a brake on coevolution. And this is especially intriguing because early in the history of life, around 1.2 billion years ago, host parasite arms races are thought to have driven the evolution of sexual recombination in the first place, since it's the genetic mixing that this allows, which fuels fast evolution to keep ahead of parasitic enemies. But millions of years ago, one enemy, cuckoo finch, seems to have lost the ability to recombine genetically in order to keep mimicry distinct. And this, as we've seen, came to have a downside in the order of hundreds of thousands of years ago, by which time hosts evolved signatures as a defense against these adaptations. So now this form of maternal inheritance is hampering the cuckoo finch, and hosts are a step ahead in the arms race. So in the case of this particular arms race played out in the long grass of Zambia, Coevolution has shaped a genetic architecture in the parasite that both facilitates and constrains an arms race, and turns out to be a double-edged sword. So in the cuckoo finch, the arms race is fought, as we've seen, through eggs, and therefore fought by females. So females produce the eggs, and so it's females' genes that are drawn to the battle. But some brood parasites mimic host chicks rather than host eggs, and this is interesting because chicks, of course, can be either male or female. So this, for example, is a brood of chicks from the nest of a Jamison skyfinch in Zambia. You'll have guessed that something must be amiss, and you would, of course, be right. Here's the cheat. It's the chick of a purple indigo bird alongside the chicks of its 5-finch hosts. In the indigo birds and also the closely related widers, the parasitic chick doesn't kill or habitually outcompete the host young, but rather is raised fairly peaceably alongside them. But looking like them is quite a challenge. So each host species has chicks with their own distinct pattern of mouth markings, skin colour, and crazy hair. And if this green-winged Martilia weren't enough to contend with, here are the mouths of the chicks of 6 more finch species occurring at our study site in Zambia, which Gabriel Jamie did brilliant work on during his recent PhD. But these chicks, of course, can be either male or female, so both sexes are drawn into battle. So here are 2 pairs of hosts and parasites photographed by him in a standardized way through a crystal prism. The 5-finch and indigo bird that we saw earlier, and the common maxwell and its parasite, pintail wider birds that I'm sure many of you will have in your gardens here at Cape Town. And as you see, they're in a perfect match. So together with a team of colleagues, Gabriel has recently shown that videos, indigo birds and widers, match their hosts not only in their visual appearance, but also in their bedding calls, illustrated by these sonograms, and even in the specific behavioral gestures that they make while they're begging for food. But the key point is that chicks come in both sexes, so both males and females need to be specialized in all of these different traits: appearance, sound, and behavior. And to achieve this, they must do 2 things. They must mate only with each other to ensure that their young look the same as them, and then they must lay their eggs in the same host species as they themselves were raised by, so the hosts are raised in the right environment. The indigo birds manage both of these things with an amazing quirk of their biology, which was illuminated through decades of careful fieldwork by Bob Payne and Lauren Payne. So growing up in host nests, both male and female indigo bird chicks imprint on the song of the host that raised them. And as adults, males then imitate the song, and females are attracted to it. So, crucially, what this means is that an indigo bird will only ever mate with another indigo bird raised by the same host as it was. And a remarkable consequence of this is that if at least 2 females accidentally lay their eggs in the nests of a new host species, these offspring will only mate with one another, and so unleash a brand new species of indigo bird in a single generation, so instant speciation. So this explains why there are many more species of indigo birds and widers than there are of their closest relative, the cuckoo finch, which they split off from around 10 million years ago. For the cuckoo finch, just like the common cuckoo, the clutch of eggs is the battleground, so only females are drawn into the arms race. And as we've seen, the female-specific chromosome is the weaponry, so only females are specialized, and this allows the cuckoo finch to stay a single species. And for the indigo birds and widers, the breed of chicks is the battleground, so both males and females must be specialized, and only complete reproductive barriers between host specialists can ensure that. So we have a flowering of indigo bird and wider species, each one loyal to its specialist hosts, but we have just one cuckoo finch, ancient and alone. It's the female's brown exterior concealing these millions of years of exquisite adaptation lying underneath. So these evolutionary battles in the long grass of Zambia have given us examples of how enemies can drive diversity in one another. So we've seen that parasites can drive hosts to evolve signatures among individual females, and that hosts can in turn drive parasites to evolve ancient strains of specialized females within species. And finally, that hosts can even drive parasites to split into separate species when both male and female parasites need to be specialist mimics. So in all these cases, egg and chick appearance has a clear genetic basis, so it's reciprocal natural selection on genes that's led to these beautiful adaptations. But now I'd like to shift gear in 2 ways. First, to consider collaborators rather than con artists. And second, to consider how interactions among species might drive another kind of biodiversity, and that's cultural rather than genetic diversity. So let's return to those greater honeyguides that we met a little earlier as thoroughly nurtured chicks. Well, as their name implies, they have a much sweeter side as well, since as adults they're involved in a unique example of human-animal cooperation. And this one arises because the favourite form of honeyguides is wax. So the West has been aware of this since 1588, when a Portuguese priest in what's now Mozambique wrote to complain that small birds flew into his church in the bush and ate his candles off the altar. What we still don't know is how honeyguides are able to digest wax, but we do know that they use our own species to get it. So these men are honey hunters from the Jawa people who live in the Niassa region of northern Mozambique, and they're searching for wild bees' nests, but they're not good at finding them alone. So as they walk, they summon some help by making a distinctive sound. Now, great honeyguides are also interested in bees' nests because they eat wax. So the birds know where the bees are but can't get at the wax, whereas the humans know how to get at the wax but can't find the bees. So the bird listens to the honey hunter's call and approaches them, giving another distinctive sound in reply, one which I'm sure many of you will know. It then flies it then calls as it flies from tree to tree, typically for several hundred meters, guiding the men to a bee's nest with uncanny accuracy. And the humans then do their bit. They gather a bundle of dry wood, they wrap it in green leaves, set it alight, and then hoist this flaming bundle high to the tree so that it billows smoke right alongside the bee's nest and subdues the bees. And sometimes, if the bees are exceptionally angry, the men have to come back at night when they're slightly more docile, owing to being cooler. So now that the bees have been subdued, the men can start to harvest the nest with their axes. And in Mozambique, this normally involves felling the entire tree. So once the tree is down, they can then chop out a rectangular access hole and haul out the honeycombs from inside. And here you can see the swarm of subdued bees gathering alongside the remains of their destroyed nest. Now it's hard work, and there's a lot of stinging in spite of the smoke. But everyone's left very pleased indeed, including the honeyguides who can then quietly flit down and eat from the waxy combs that have been left behind. Now it's often believed that honeyguides also guide honey badgers, but there's no evidence that this ever happens, so it seems that humans are the honeyguides' only mutualistic partners. So the key point is that this human-animal mutualism exposes the wax for the honeyguides and the honey for the humans. So the bird exchanges its knowledge for the human skills to mutual benefit. And the use of fire and tools are skills which our hominid ancestors could have provided to honeyguides for hundreds of thousands of years. So mutualisms are everywhere in nature, but what sets this one apart from most others is that our own species is half of it. So this means we're influenced by variation in human culture. So honeyguides probably aren't interacting with species or individual traits as we saw in bird parasites, but rather they're interacting with cultural traits that vary among human populations but are consistent within them. And this might be a situation that's more broadly relevant beyond this particular mutualism, because learned cultural traits are quickly emerging as widespread in nature in animals from great tits to sperm whales. And we might expect this to drive reciprocal diversity in the species that such animal cultures interact with. So does variation in human culture have consequences for how this mutualism unfolds? Well, in many places, human culture has in fact halted the mutualism altogether. People often rely on beehives, or aren't much interested in honey because they can more easily buy sugar instead. So in large parts of Africa, such as much of our own country, honeyguides have largely given up trying to guide us. They seem to get by without us, although they do seem possibly less abundant in such places. But even within the same part of Africa, different coexisting groups of people aren't all of use to honeyguides. So for example, in Tanzania, the Hadza people, who are superb honey hunters, live alongside the pastoralist Datoga people who are very wary of bees and prefer to crave for honey with the Hadza. So it should be a honeyguide to tell them apart. And third, even within a single honey hunting culture, people differ in their levels of skill and motivation. Most obviously, nearly all honey hunters are men, although I love this photo of an honourable exception, our colleague Mama Fatima Barasani in Mozambique, who's a demon honey hunter. Although very much an exception. And once again, we should pay honeyguides to tell people apart. So perhaps it's no surprise then that humans actively signal their utility to a honeyguide by specifically advertising that they're looking for a honeyguide with whom to cooperate. So here's a natural interaction starting in Mozambique. It's really wonderful to have your study species come to find you, as humans probably been, as honeyguides probably been doing for as long as we've been human, and possibly for even longer. If you ask the honey hunters why they make this particular brr sound, they'll say, well, obviously it's the best way to attract a honeyguide, because honeyguides understand. So to test this claim, two honey hunters and I did a simple experiment in the Niassa National Reserve in Mozambique, where I work with the amazing support of the conservationists Colleen and Pete Begg and a wonderful team. And we had the honey hunters and I played back different sounds on 15-minute walking transects, simulating short honey hunting trips. Either an animal control sound, which was the sound of the turtle dove, which you all know well, or a human control sound, which were arbitrary human sounds, such as the honey hunters' own names, or brr. So what we found was that compared to these control sounds, going brr, which is the grey box on the right, doubles your chances of being guided by a honeyguide. It triples your chances of actually finding a bee's nest, since the honeyguides are less likely to give up on you while you're following them. So this experimentally validates the honey hunters' own claims that improves their foraging success both in attracting honeyguides and in maintaining the birds' enthusiasm to keep cooperating with them. So this communication is in fact reciprocal. Honeyguides tell us where the bees' nests are, and we tell honeyguides that we're the best partners to help them to get the wax. The brr is only used in the far north of Mozambique, and elsewhere, different cultures use completely different calls when seeking and following honeyguides. For example, the Hadza, people of the north of Tanzania, who are also expert honey hunters, make this sound in exactly the same context. So this seemingly arbitrary variation among cultures needs to be learned. And the Jawa and the Hadza tell us that they learn these calls from their fathers, and no one diverges from that cultural tradition because they'll find less honey. Now, my wonderful colleague Brian Wood is an anthropologist who's worked with Hadza for a couple of decades, and he and I wondered whether honeyguides learn these cultural traditions too. So together we did an experiment to see whether honeyguides know the signals of their local human culture, but not those of a foreign culture. So we could either swap honeyguides among humans or humans among honeyguides, but the second of those is much easier. So we took human recordings from one country to another. So first, we again walked transects in Mozambique, but this time the treatments were either an arbitrary human sound or one of two kinds of honey hunting signals, the Jawa brr or that Hadza melodic whistle. And then Brian and I did exactly the same thing with the Hadza in Tanzania. This is one of those slides where it seems almost indecent to compress so much effort into such a simple result. So however, developments onwards, this is what we found. So in Mozambique, honeyguides again preferred the local Jawa brr signal above all. That's the green bar on the right, with nearly 3/4 of transects yielding a guiding honeyguide. However, in only a quarter of transects accompanied by a Hadza whistle, that's the orange bar on the left, did a guiding bird come to us. In fact, this was even worse than the arbitrary human sounds in blue, which were in between. In Tanzania, by contrast, we found the exact opposite. We found that the Hadza honeyguides were very interested in the Jawa brr, they're again in green, which only drew in a honeyguide on a quarter of transects. But we were guided on 82% of transects accompanied by that beautiful melodic whistle used by the Hadza themselves. So what this tells us is that there isn't anything about these honey hunting calls that's intrinsically attractive to a honeyguide. Rather, honeyguides seem to know and prefer their own local human culture. An alternative hypothesis could in fact also explain these findings if the form of these signals is precisely tuned to the sound transmission properties of their respective environments by a process called sensory driving. So perhaps honeyguides aren't choosing the local call, it's just that they can hear it better. And if that's the case, then we should expect sounds to attenuate faster in a foreign environment than in their local environment, which we were able to test through some very tedious recordings happening here. And what we found was that, in fact, each sound type attenuates very similarly in both environments. So each of those kinds of boxes are from Mozambique and Tanzania, and as you see, they show very, very similar sound volumes. So our results are best explained by the hypothesis that honeyguides know their own local culture. And I'd like now just to briefly return to those experimental findings to touch on two further interesting points. So first, it's striking that the Hadza honeyguides were 3 times as likely to respond to a local signal versus a controlled human sound, whereas Jawa honeyguides were only half again as likely to do so. And this makes sense because the Jawa are the only cultural group in that landscape, and just about all Jawa men know how to harvest the bee's nest. Whereas by contrast, the Hadza share the landscape with the Toga pastoralists who are frightened of bees and no use to a honeyguide. So since Hadza honeyguides face more variation in the quality of their potential partners, we should expect them to be more choosy about whom they cooperate with, and indeed that's exactly what we see. Well, second, in Mozambique, these foreign whistles, in fact, tend to fare worse than arbitrary human sounds. So this suggests that the Hadza whistles may not even act as a cue that a human is present in the environment. Whereas by contrast, in Tanzania, the foreign Reciprocal fared similarly badly as arbitrary human sounds, perhaps because at least the Reciprocal, being a vocal sound, is at least recognised as human. So why might the Hadza not want to sound human? Well, it's possible that this whistle signal minimises trade-offs between different kinds of foraging. So when out in the bush, the Hadza aren't only looking for honey, they're also looking for animals which they kill with bows and arrows. And for this, stealth is essential. So perhaps it's better to sound like a bird to all animals, except the one species who's heard what it is, who's learned what it is that you mean. So how do honeyguides acquire that understanding of what humans mean? Well, since honeyguides are raised by the species, they clearly can't learn from their own parents. So we suspect that instead young honeyguides speed up learning by watching other honeyguides via a process called social learning, and so are quick to fine-tune their behaviour when the human environment is in their favour, but also quickly drop it when the human environment isn't, especially when humans aren't interested in honeyguides as partners, but perhaps as prey animals. So perhaps this may lead to a mosaic of honeyguide cultural traditions that map onto those of their human partners. So we're currently testing some of these ideas, in part thanks to the efforts of 20 honey hunters, 18 men and 2 women, who have for the last 2 years, 2.5 years now, been recording all of their usual honey hunting trips for us. And they've done so with these rugged devices, essentially souped up cell phones, which run a custom app, which allow it to be used solely via icons. So this allows accurate data collection, even by those who are unable to read. Here I'd like especially to thank David Lloyd-Jones, who's been absolutely instrumental in this project, as well as the amazing help of the app database Habitat Info. So honey hunters are GPS tracked throughout, and they use the app to indicate when they start calling for a honeyguide and when a bird begins to guide them. So these red dots are all such places where a guiding event began. And these green dots show where the honey hunters found these nests and used the app to record how much wax they leave behind and how much honey they harvested, which can be up to 40 kilos from a single nest. And it all adds up to look something along these lines at a larger scale. So far, the hunters have recorded over 1,000 honey hunting trips involving over 5,000 bees nests that have used honey for hunters and wax for honeyguides. So we're just starting to tackle this data set to ask exactly what the costs and benefits are for each party of this specialised channel of communication and cooperation. So just to start then, the experiments that I've just shown you show that in these two interacting species, communication goes both ways. Honeyguides tell us where the bees nests are with amazing accuracy, and we in turn tell honeyguides as a partner to help them get the wax. And more broadly, what it implies is that a wild animal can correctly attach meaning and respond appropriately to a human signal of cooperation. And although this is well known in dogs and in other human foraging partners such as falcons and cormorants, these all involve trained or domesticated animals who are specifically taught, or in some cases even coerced, to cooperate, whereas What's remarkable about the human-honeyguide relationship is that it involves free-living wild animals whose interactions with us have evolved through natural selection. But this is coevolution. In a strict genetic sense, perhaps not, since while our human or hominid ancestors almost certainly influenced honeyguide evolution by facilitating their access to wax, we can only speculate about whether honeyguides reciprocally influenced our own evolution, as genetic coevolution would imply. But perhaps learned traits are reciprocally influencing each other in a way that's akin to genetic evolution. So once honeyguides have learned to respond to these specific signals that humans give them, reciprocally it doesn't pay any human to use any other sound, because they'll find less honey, just as the Joel honey practices tell us. So perhaps cultural coevolution could lead to a mosaic of honeyguide cultural traditions that map onto our own. So next we need to find out whether learning has led to culture in both species by trying to test whether honeyguides learn socially from other honeyguides, which is a hallmark of animal cultures. Meanwhile, in the long grass underneath these bees' nests, the prionies are nest building, and their egg patterns continue to evolve in response to the egg patterns of cuckoo finches, which reciprocally evolve in return. And just as we can swap eggs among nests to unpick these hundreds of thousands of years of genetic evolution among enemies, so we can swap people's voices among different parts of Africa to test how generations of learning have eased cooperation among collaborators. Now, some have argued that our greatest challenge as biologists is to understand how evolution continues to shape the web of life during a time where we are altering its component parts with unprecedented speed. So perhaps in so doing, we need to consider how multiple forms of biodiversity might influence how species interact, and so how they cope in this very rapidly changing world. And no species, of course, is easier to experimentally manipulate than our own, which we hope will allow us to work out how genes and culture together help to keep this honeyguide human mutualism alive and stable in those parts of Africa where this part of our own evolutionary history still unfolds. I'd like to stop here with gratitude to our funders for their amazing support, and warmest thanks to my own many mutualistic partners, further to those who are already highlighted. Above all, the Chola community in Zambia and the Nyasa and Hadza communities who welcomed us with such generosity. And special thanks to the honey hunters who cheerfully put up with walking many hundreds of k's with us, making the wrong noises. And thank you very much for perhaps got time for a couple of questions, if any of you have any.
Has there been any individual variation between the honey hunters and the honeyguides, that they recognize which person is one?
That's a great question, and we're trying to answer it at the moment. There is, to answer the first part of your question, some variation, but there are certain persistent features that do remain constant, but everyone has their own flourish, if you wish. And that could indeed provide a cue to honeyguides of being able to refine the same individual who's been generous to them in the future. It's not an easy question to address experimentally, but we're doing our best. So perhaps I'll have an answer for you in a year or two. I should emphasize that this is a vast area. You may have seen on the map the scale bar of 10 Ks, and there's thousands of birds operating in that area. So it's